Hey biology students, this is our second lecture video on the topic of viral diseases. I have a message for you written on the screen. Can you read it? Does it not make any sense? Would you say that this message on this screen is perhaps anti-sense? Oh, you read it. Okay. It's backwards though, isn't it? Well, this is a message for you. You're very close to the end of the semester now, and you've made it through a course that's not designed to be taught online, which is microbiology. And so my message to you is written here backwards because I want to give you an analogy to RNA viruses like influenza, which is our topic in this video. So these are RNA viruses where the genome is what we call antisense or negative single-stranded RNA, right? So we use this term antisense, and we write this as a negative, which means antisense single-stranded RNA. So we would classify influenza as antisense single-stranded RNA. So that means that the way the RNA is aligned does not make sense to the cell. Just like if I wrote this message and you're used to reading from left to right, and if you read it from left to right, it says snoitalutarganok. Well, what in the world does that mean? Well, it means nothing. Okay, it's antisense or nonsense, right? So the point is that these RNA viruses that are antisense, they have to produce a sense copy of their RNA genome in order for that RNA to be read by the cell as messenger RNA. So what has to happen is we need to make a version of this message that can be actually understood. So let's write it the way I intended it to be written. With an explanation point. Congratulations. You are almost at the end of the semester. Hang in there. These are our last two lecture videos. So this influenza virus is going to have to convert its genome into sense RNA, which can be essentially understood by the cell as equivalent to messenger RNA. So let's talk about that. Okay, so first of all, the structure of influenza. It's enveloped and it's single-stranded RNA. However, it's what we call a segmented genome. A segmented genome means that there are multiple copies of the RNA. In fact, there are 8 copies of the RNA. So let's do a diagram here of just a general picture of influenza. So it's enveloped with a capsid, and in there we're going to have 8. Don't have quite enough room, but 8 little squiggles, okay? Inside there, that would be the 8 copies of its antisense single-stranded RNA genome. Now, along the edges of this enveloped virus, this influenza virus, we're going to see various glycoprotein spikes. And so for influenza, these glycoprotein spikes are called hemagglutinin, or HA, or sometimes just H. Now, there are actually 18 different H's. So there's H1, H2, all the way up through H18. So there's some version of the hemagglutinin protein on the surface of our virus. So we might have something like H3, right? So we might have H3 as a spike along the surface of the virus. And then there's a second spike, and the second spike is called neuraminidase, or sometimes just referred to as N for short. Okay, and there's recognized to be 10 different N proteins. So there can be N1 or N2, all the way up through N10. So let's say this is N2. So we'll put the N2 on the surface. So this is a little spike, right? Okay, so what's the name of this virus? Well, the way we name it is based off of which version of the hemagglutinin and which version of the neuraminidase spike. So this virus would be called H3N2. So if there are 18 different hemagglutinin protein types and 10 different neuraminidase protein types, how many different combinations of the H and the N can we see? Well, that's right. It would be, you would multiply 18 by 10, and we would get about 180 different combinations. Now, that's just of the H and the N protein. Something else to keep in mind is that all influenza strains are also named based off of what strain of influenza, where there are 3 recognized strains. So we call them the A type, and the A type is the most common. It also causes the most severe disease. So these are found in humans as well as in swine as well as in birds. And most of the time, these viruses do originate in birds. So we see a zoonotic spillover event from an animal into humans. And when that does happen, that is what's responsible for pandemic strains of influenza. Strain B, there's a little bit less versions of strain B influenza. They all infect humans. Strain C can infect humans, dogs, and swine, although strain C causes a more mild disease. Okay, so influenza binds to sialic acid on the surface of ciliated respiratory epithelial cells. So here's influenza shown with its H spike, which is the attachment ligand. The virus enters by receptor-mediated endocytosis and enters into an endosome. The virus is uncoated and releases its 8 segments of single-stranded RNA, which enter the nucleus of the cell. Recall, this is antisense RNA, so the virus relies on RNA replicase, an enzyme that is prepackaged with the influenza virion. The RNA replicase facilitates copying the RNA into positive sense RNA, which is the mRNA that can be used to direct the synthesis of viral proteins in the ribosome of the cell. So here we're going to show capsid proteins, H proteins, and now I also want to draw in the N protein. Let's use green. So the green spike protein is the N spike, or neuraminidase. The neuraminidase is also important in the life cycle of the virus. It's important in the release stage because as the virus buds from the cell, it is stuck to the sialic acid. And it's the function of neuraminidase to cleave the sialic acid and allow for release of influenza virus. Now let's talk about the stages of this life cycle that can be inhibited by medications that are used to treat influenza. So there are 4 well-known prescription medications that can be used to treat various stages of the life cycle of influenza, and they are grouped into two categories. One is neuraminidase inhibitors. Remember, again, neuraminidase is the enzyme, the N spike, that allows the sialic acid to be cleaved and allows for the release of the virus. So without neuraminidase, the new viruses would not be able to be released. So that's why it's an effective target as an antiviral. Okay, so these would all be, again, antiviral. So the three that are in this category are Tamiflu, Relenza, and Peramivir. The second type of medication is an RNA replicase inhibitor. Why would that be an effective target? Well, RNA replicase is the enzyme here shown in purple that is responsible for copying the antisense genome into sense genome and then recopying to make more antisense genome to be packaged with the virion. And by the way, RNA replicase is part of this package of the virion. It comes inside the virion. So we should actually be showing that in my picture. And we'll make RNA replicase here. We'll make it a little purple dot. Okay, I know I'm showing you here like purple Pac-Man, but this will be RNA replicase here in purple. So it would show up inside of the capsid. So let's put a little RNA replicase in there, okay? It's important to make sure that that's in there because otherwise the virus would be totally out of luck. It would uncoat and release its antisense genome and nobody could read it. So RNA replicase is the reason why those viral proteins can be translated in the cell. So if you take that guy out with an RNA replicase inhibitor, that is a very effective treatment. And that treatment is called Xofluza. This one was most recently licensed in 2018. So these are some effective antivirals that can be prescribed when we have patients who have severe influenza. So now let's look at a swine cell. Swine cells can be mixing vessels for new strains of influenza. So let's say we have this influenza strain in purple, and we have this influenza strain in red. Both co-infect the swine cell. We'll say red is human-derived and purple is swine-derived. So now we have RNA from both viruses inside of the swine cell. And this is a process we call antigenic shift. Genetic recombination results. The new influenza that results from this infection has RNA from both types of influenza. Purple proteins and red proteins result. This is a new strain of influenza. This is one of the big advantages of influenza, which is that you can get your annual flu vaccine. And who should get the flu vaccine? Everybody should get the flu vaccine, unless you are an infant under 6 months old, okay? So the flu vaccine, especially the inactivated. So when we talked about vaccines, we talked about different types of vaccines. So the inactivated flu vaccine, this is the one that's the flu shot. Who should get the flu shot? Everybody should get the flu shot, okay? The only ones who really can't are infants under 6 months old. Otherwise, everybody else should be able to get the flu shot, including pregnant women. Now, there is also, so we'll say everyone, the exception, babies less than 6 months old. So how are we going to protect the babies that are less than 6 months old? Vaccinate the caregivers. Okay? Remember, when you get vaccinated, not only are you protecting yourself, but you are protecting everybody around you, including those babies who are not old enough to get the flu vaccine. By vaccinating the caregivers, the parents, the daycare workers, you are protecting that baby. Okay, so it is protecting yourself, but it's protecting those who cannot receive the vaccine. Now, there's also a live attenuated flu vaccine. This one is a nasal spray. Because it's live attenuated, recall this would not be a good choice for a pregnant woman or for someone who has an immunocompromised state. They should get the inactivated flu vaccine. So the flu vaccine is usually given as either what we call trivalent or quadrivalent. Now, this refers to the number of flu strains in the vaccine. So tri means 3, so you could imagine there would be 3 different influenza strains in the one vaccine. And quad means 4, so there would be 4 different influenza strains in one vaccine. Here's the vaccine that was given in 2019. It's called Fluzone. So this is the 2019 inactivated flu vaccine. It's quadrivalent. And I want to talk to you about how we name these strains. So what happens every year? Well, the World Health Organization, we call them the WHO, okay, the World Health Organization gets together every year, usually in about February, and there are locations, there are 5 labs across the world. They collect flu specimens from people who are infected. And these labs are located in the US, China, Japan, Australia, and the UK. Okay, so the World Health Organization gets together with these 5 different labs and scientists from those 5 countries, and they get together, and it's really a very small group. It's like 12 to 15 people, scientists, get together, and they make their best guess based on infection rates that they are seeing at their hospitals in their respective countries about what strains of flu are circulating the globe. And they make their best guess, and they put those strains in the flu vaccine. Now let's talk about how flu virus is named. It's named by the type of influenza, the location or origin of the sample, the strain number, the year that strain was isolated, and the H and the N subtype, for type A influenza only. So these are the strains that are in the Fluzone quadrivalent vaccine for 2019. Notice that for the type B virus, we do not include the H and the N subtype. The reason is that the B virus, there are fewer types, and we do not need to further subclassify. Usually, we also put these slash marks in between each designation of the name of this particular viral strain. The last thing I'd like to do is compare influenza with COVID. So what's the same? Well, they both cause a respiratory illness with very similar symptoms: fever, cough, nasal congestion, sometimes nausea and vomiting, and body aches or shortness of breath. To differentiate, we could do an RT-PCR to test for influenza viral RNA compared to coronavirus viral RNA. We talked about that test in the last lecture video on coronavirus. The pathology of the two are very similar, with variable results from mild to severe, including pneumonia and potentially acute respiratory distress syndrome. For influenza, usually there is a secondary infection with bacteria. Transmission is the same, either droplet or fomites. The incubation period for influenza is 1 to 4 days, and the person is contagious for 3 to 14 days. For COVID, the incubation period is between 4 to 14 days, but up to 24 days, and contagious about the first 18 days of the infection. Case fatality for influenza is less than 1%. For COVID, it's unknown, but approximated to be 1 to 3%. The treatment for influenza, there are 4 prescription antivirals. For COVID, all treatments are experimental. Ventilators, as well as some anti-inflammatory medication, has been shown to be somewhat effective. Prevention for influenza, the annual flu vaccine. You want to get that every year right around Halloween before the winter season sets in. So think about that next time it's Halloween. What's scarier than Halloween? Not getting your flu vaccine. What about COVID? That's right, there's no vaccine, but there's the potential for a vaccine hopefully within the next year or so.